Good day, and whatever day it is, and welcome to the fifth day of this series of lectures on the genetics of the immune system. So what I'd like for you to do as we get started here is take out your genetics of the immune system study guide, and we're going to pick up where we left off last time. And if you remember, last time I asked you to go ahead and download uh, the second study guide called ABO blood types, an example of multiple alleles, and it was a self-guided sort of self-teaching packet that I asked you to work through, and it taught you about the most famous example of multiple alleles in humans, and that is the inheritance of ABO blood types. And I hope that working through that study guide made sense to you. If you have any questions at all, please email me, and I'll do my best to answer your questions via email. But in the meantime, I'm going to I have placed into the module the genetics of the immune system module. I've put in there the answer key for the ABO blood types study guide, so you can download that and compare the answers to the answers that you came up with as you worked through that study guide. So I think that'll help a great deal. But what I want to do today is we're going to go ahead and finish this study guide, and so this will be the fifth and last lecture that has taken us through this entire study guide. And in the next couple days, you'll be doing an online activity on ABO blood types and ABO blood typing that involves a game as well. And I think that will even help you more in your understanding of how ABO blood types are inherited and how transfusions between donors and recipients are matched up, and what role the immune system plays in all of that. But what I want to do today is look at, in your Genetics of the Immune System study guide, page 14. So go ahead and turn to page 14 in your study guide so you can fill that out as we go along, using that and what you see on the screen here. So what we're going to look at today at the top of page 14 is another example of multiple alleles, and this is called HLA types. Now, I know that the classic example of multiple alleles that we talk about in humans is ABO blood types involving 3 different alleles, but as the expression goes, you ain't seen nothing yet. When you look at the inheritance of HLA types, which we're going to look at today, this is a major example of a trait that is controlled by multiple alleles. And this is what makes it so difficult in trying to match up donors and recipients when we're talking about tissue or organ transplants. I know a couple different guys who have had kidney transplants, and believe me, they were on a waiting list for years, and it took a long time to find a match that would work. And so what I'd like for us to do is look at question A at the top of page 14 in your study guide, and I want you to think about the answer to that question. What is the biggest problem that must be overcome in tissue or organ transplants? And so I want to give you a few seconds to think about that. You can even pause the video if you'd like to give it some thought, and then afterwards restart the video, and we will compare our ideas on the answer to that question. All right, if you said rejection, you are absolutely right. I know a little boy who had to have a kidney transplant, and the first time, it was very heartbreaking. The first time he received a kidney, his immune system started to violently attack it. because the antigens all over the cells that made up that kidney that was donated to him were so foreign to his immune system. His immune system freaked out, started to attack and kill the kidney because of all the antigens on the surface of that donor kidney that were just too different. The genetics of the donor were just too different from the genetics of the recipient, that the immune system violently attacked and rejected and killed that kidney. So it didn't work. Fortunately, a couple years later, a closer match became available, and it's kind of a very heartwarming story. It was actually the little boy's elementary teacher, class teacher that he had at the time, who was a match and who donated one of his kidneys to his student. And as far as I know, the last I've heard, the little boy is doing fine. The kidney's working just great, and there's a happy ending to that story. So, biggest problem is rejection, and so I want us to look at why transplanted organs are often rejected. So take a look at section B on page 14. There's a real kind of a simplified diagram that I've put there on page 14 of chromosome number 6. And there's a group of 4 genes up there on the P arm of chromosome number 6 that control the manufacture of certain antigen molecules that are on the surfaces of our body cells. They're on the surfaces of all the cells that make up our body. And they are called, these antigens that are produced by these genes are called human leukocyte antigens, and you can see that on the diagram on page 14. Human leukocyte antigens. And the reason they're called human leukocyte antigens is leukocyte is a fancy word for white blood cell. And these antigens were first discovered on the surfaces of white blood cells. But like I said, they're on the surfaces of all the cells of the body, we now know. And human because, well, duh, that's what we are. So human leukocyte antigens, those are specific antigen molecules on the surfaces of our body cells. I've got them all over the surface of my body cells. They're like little name tag molecules that say, "Hi, I'm Mr. Ruhl." And so my immune system encounters those antigens all the time. My immune system kind of says, "Hey, those are friendlies. Do not attack." And that's a good thing. Now, these 4 genes that you see on the P arm of chromosome number 6, human leukocyte antigen, or HLA-A, HLA-B, HLA-C, and HLA-D, which actually contains 3 subregions, HLA-DP, DQ, and DR. So we're looking at a total of 6 different genes here, clustered in a group on the P arm of chromosome number 6, that all produce these protein molecules called antigens on the surfaces of our cells. And those 6 genes make up a group of genes there on chromosome number 6 called the major histocompatibility complex, or MHC. Major histocompatibility complex. Major because they're very important when it comes to trying to match those genes up with the genes of donor and recipients. When we're talking organ transplants, they're major. Those 6 genes have to be matched up as closely as possible when we're trying to find a donor that matches a recipient. That's why it's called major. Histocompatibility. Histology. In biology or medicine, histology is the study of tissues. Histo has to do with tissues. So histocompatibility, we're talking about tissue compatibility. How compatible is the genetics of the donor compared to the recipient when we're trying to match them up? And complex just means it's a group of genes. This group of genes here on the P arm of chromosome 6 that must be matched up as closely as possible when we're trying to match donor and recipient. Okay, so that's kind of a simple diagram on page 14 of that chromosome number 6. Now keep in mind, you've got 2 chromosome 6s, so that means your HLA antigen gene, HLA-A, you've got 2 copies of that. Remember, your genes come in pairs. And your human leukocyte antigen A allele on one chromosome 6 very likely will be different than the one that you have on your other chromosome 6, because again, these genes come in pairs. Now, take a look at the screen here in the presentation. This is a more accurate diagram of chromosome 6 showing HLA-A, HLA-B, HLA-C, and then the 3 different subregions of HLA-D, DR, DQ, and DP. And you can see it's kind of, the view is kind of zoomed out or exploded out. And you can kind of get the idea here from this diagram, I think, that those genes are all clustered very close together in a very small region, about almost right in the middle of the P arm there of chromosome 6. And that cluster again is called the major histocompatibility complex. Now earlier I said the inheritance of these HLA types represent a major example of multiple alleles, and I'm talking major here because, for example, take a look at this diagram on the screen. The number of different alleles that we humans have for HLA-A is 349. Now, at least as of the last time I was reading and putting together this presentation, 349 different kinds of alleles. So we're not talking about big A versus little a. That would only be 2 kinds of alleles. We're talking 349 different kinds of HLA-A alleles. See what I mean when I said this? The inheritance of HLA types is a major example of multiple alleles. And then check this out. HLA-B, there are 620 different kinds of alleles for that particular gene. So, like, for example, on one of my 2 chromosome 6s, I may have HLA-B type number 600, and you might have HLA-B type number 335, but there are 627 different kinds. Can you see why it is so difficult if somebody needs, let's say, a new kidney, to find a suitable donor? My gosh, there are 627 different HLA-B alleles, and there are 349 different HLA-A alleles. We've got to try to match these up as close as possible. And then if we look at HLA-C, there are 182 different kinds of HLA-C alleles. So I think you can begin to see just why it is so difficult trying to match up donors and recipients. Now, fortunately, if somebody needs, let's say, a heart transplant, the doctors and the geneticists will do genetic testing on that person to determine that person's specific kinds of HLA-A genes HLA-B genes, HLA-C genes, and the 3 different groups of the HLA-D genes. They'll figure out the specific genotype, what alleles the person has for each of those 6 genes, and those will be kept in a database in a computer. And fortunately, these databases are connected all over the country via the internet, connecting hospitals all over the country. So if somebody, let's say, needs a kidney and they live in Indiana, and a potential donor dies, let's say, in a motorcycle accident in California, and he had indicated on his driver's license that he wanted to be an organ donor. If that were the case, that victim of that motorcycle accident would be taken to the hospital, the organs removed and kept on ice, kept cold to keep them, and they would be genetically tested to test the specific HLA types of the genes of that unfortunate donor, and put into the computers and databases matched all over the country. And if that person in Indiana who needs a kidney, if it turns out to be a match, the match of the kidney of that donor who died in the motorcycle accident would be quickly shipped on ice, kept cold, to the hospital in Indianapolis. The recipient or the recipient's parents would receive a text and told to get to the appropriate hospital in Indianapolis where the transplant surgery would take place. That's kind of how it works. Now, take a look at the top of page 15 in your study guide, and let's answer that question there. Letter A at the top of page 15, how many different alleles are there for each locus, each gene location, for each of these 3 HLA alleles that we've looked at here? Well, I think the answer you could put is a bunch. And letter B says, with so many different possible alleles at each locus, each person has a different overall HLA type called a blank. The answer there is haplotype. 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 So if you needed an organ transplant, they would do genetic testing on you to find out your HLA antigen genotype, which is called a haplotype. And your haplotype would be made up of a listing of the specific HLA-A, HLA-B, HLA-C, HLA-DR, HLA-DQ, HLA-DP. The specific alleles that you have, it would be a listing of the specific alleles that you have, and that would be your haplotype. So, like, for example, your haplotype might be HLA-A number 200, HLA-B number 3, HLA-C number 119, and so on. And if you needed an organ transplant, you see that would be put into the database, which would be made available over the internet. So this is kind of how it works. Now, letter C on page 15, in tissue or organ transplants, doctors try to match up at least 4 of those 6 genes. Among unrelated donors and recipients, the chances for a successful match are about 1 in 100,000. So you can see why the technology, computer databases, the internet, the interconnectedness among hospitals all over the country, you can see why that is so important. Now, take a look here on the screen and also at the bottom of page 15 in your study guide. And what we're looking at here is the inheritance of HLA haplotypes in a family. And so if you'll look at the two parents here. Remember, squares are males and circles are females. Look at the two parents of these 4 children. Dad, look there. There's Dad's haplotype. There's his haplotype. Okay, this is one way of representing it. He has HLA-A A1 and HLA-A3. Remember that Dad's got 2 chromosome number 6s. Maybe he received A1 from his mom, maybe he received A3 from his dad, or vice versa. So Dad's HLA haplotype we would read like this: A1, A3, B8, B18, C4, C2, D2, D1. And then you can look over to the right and you can see what mom's haplotype is. I think if you look at the inheritance of this, look at their 4 children down there. Look at the firstborn daughter on the left-hand side. Notice she got one of those chromosome 6s with the group of 4 genes that make up the major histocompatibility complex. She got one of those chromosome 6s from one parent there. The A1 and B8 and C4 and D2 genes. Who did she get that particular chromosome 6 from that contains A1, B8, C4, and D2? Take a look at the pedigree in detail, think about it, and then we'll come back and we'll discuss that. Okay, you should be able to see that she got that particular chromosome 6 from her dad. And then the bottom one that she has, A6, B27, C1, D5, remember, that is the group of 4 HLA genes that she got from mama. Okay? And then you can see the other siblings too, and you can trace back to see which parent the siblings got their particular chromosome 6s from. I think you can see in this diagram why the first place they'll look for possible donors will be in close relatives, family members. And then, of course, the more distantly you are related to a possible donor, the less likely you're going to have a match. And I think you can kind of see that in this family right here. The more closely related the recipient is to potential donors, the more likely it is for a successful match. But it doesn't always work. Sometimes just by chance, they'll find a even better match out there in the world among some unrelated person. So I hope you can kind of appreciate through this discussion why it is so difficult to find a successful match when we're talking about organ transplants. And it all has to do with the fact that these HLA antigen genes, part of the major histocompatibility complex, are a major example of multiple alleles. There are so many different possible kinds of alleles for each of these HLA genes, making it very difficult to find an exact match. It's a whole lot easier to match up blood types if somebody needs a blood transfusion, right? Because there aren't that many different kinds of blood types. There are only 3 possible alleles, and that drastically reduces the number of different kinds of blood types. So, believe it or not, now we are finished with this Genetics of the Immune System study guide. And I'll tell you what, you do not have to hand this in. But I'm going to do something here that I've not done with you guys all school year. This is going to be our last test of the semester. And it's not going to be a quiz, it's going to be a test. So it will be worth 100 points. And when we get around to taking that test, feel free to use, I know this is going to blow you away maybe not if you hear this, please feel free to use this study guide that we just finished when you take that test. So we're going to call it an open test, or excuse me, an open study guide test. And I figure I can do this since I'm the only one teaching this course. I can do whatever I want, right? And this is just something I've decided to do. So, Merry Christmas, or maybe a late Christmas gift or early Christmas gift, however way you want to look at it. And before we do have the test day on this study guide, though, we're going to be doing a couple different online activities. One of them is this blood typing virtual lab that I told you about towards the beginning of this lecture today. And the other thing we're going to be doing, because it's very timely and relevant, I think, is we're going to be watching online a COVID-19 update with some questions that you can answer while you're watching that particular video. And I think it'll be relevant and important to you for a couple different reasons. Number one, you'll be, by the time you do that activity online, you'll be kind of an expert on the immune system and the genetics of the immune system and how it works. And then the second reason I think you'll find that activity interesting and relevant is because it deals with what we're going through as a world right now, this COVID-19 coronavirus pandemic. And I think you'll find that to be very interesting. So that's the end of this study guide for today. And like I said, you do not have to turn this study guide in, but when we take the test on it, you will be able to use your study guide as you take that test. So that's the end of the lecture for today. I'm going to say goodbye for today. Have a nice day. Stay safe. And we'll talk to you later. Bye-bye.